Everyone. I guess some of you may have heard our presentation before, but for those of you who are new to us, thank you for your time. I'll just start, again, we also have a forward-looking statement. We are a very dedicated team of people who really want to make a difference by much better diagnosing cancer to make much better outcomes. So we start with this. People say, what, your facility, why start with that? This is our facility in Belgium. It shows in the last 3 years, we've got it very serious about doing a lot of background work on our platform. We've always believed, it's been 10 years since the company was founded by myself and a couple of the scientists, that the epigenetic signaling was as important as the genetic in diagnosing cancer and a range of other diseases. And I'll get to what that means in a minute. We started off very small. We're now quite a large company for this kind of space, and we've achieved an awful lot, and I think we've got a lot more to go. This facility, people say, why Belgium? As an American company listed and being Australian in England. We paid 1.2 million euros for that facility. It's 20,000 square feet, and it now holds over 30 scientists and other people working there. And it just shows, if that was in Cambridge in England or in Cambridge in Massachusetts, we'd pay that much per year at least. We've managed to do a massive amount in this space with a very small budget, which I'll get to next. So it's very important to us, and because we're in the Walloon region of Belgium, we've also had a very large amount of regional funding, $5-7 million now. Also, our trials are massively subsidised by the American government, by European governments, and by Asian governments, which I'll get to in a second. So I'll just start with some of the financials. Being an American company, this is where we start. VolitionRx is our symbol. Our market cap, actually that's a little out of date. We've had a very good month. It's about 225 now. We've had a large range. As I said, we did a lot of background work over the last several years on the platform itself. And while that happened, the market did decrease, but it's roared back again. We are very thrifty for company doing as much work as we're doing, fundamental work, as well as a range of product work and a very large number of trials, the larger of which are over 10,000 people. And again, very heavily subsidised by governments around the world, because they realise the only way of dealing with cancer beyond actually curing it, which unfortunately it's a tricky beast, is diagnosing early and resecting the cancer in stage 1 or stage 2. So just a quick summary. We are, as I said, a company doing a range of blood tests under development. We believe the only way of making a real screening test is at least 80 or 90% of people actually do it. There are quite a few screens out there for different cancers. Some are accurate, some are very inaccurate. The problem with all of them, that aren't blood tests and routine blood tests, is people just don't take them. And the accuracy in those people is very easy to work out. It's exactly 0, and it's a real tragedy. So the only way of doing it is a truly routine blood test that's part of your normal blood work that's low cost. And that's what we're doing. And there are very, very few groups, any other opportunities apart from nucleosomes that are truly routine. We've had some great proof of concept data in a range of cancers. We're now doing large trials. The nucleosome, the structures we're after, are also preserved through a wide range of species, including dogs and things like yeast, in fact. So it's a very, very broad platform. And we've also been capturing the nucleosomes, which I'll get to later, explain what that means. Colorectal cancer has been our lead indication. We've got very large trials going on 3 continents now. And we've also had some very good work in lung recently. So we have 7,000 patients in Taiwan being collected for colorectal, over 1,000 in lung. We have about 10,000 in the US and a range of trials in Europe. But the changes we're after are pan-cancer. They seem to be in a very, very wide range of cancers. And again, I'll get to that in a minute. So we're in Texas at the end of this month. We're working with Texas A&M to help develop the products in the animal market. And we've had ongoing studies with Oxford in endometriosis. And we're selling research kits for looking for the same markers for a range of other things. So again, like the previous speaker, I'm not a scientist, but I'll just walk you through the science briefly. Some of you are probably familiar with the chromosome. The DNA, if you unwrap it, every 200 base pairs or thereabouts, there's one of these, which is a nucleosome. The yellow, which is the DNA, is the genetics. This is the epigenetics, which is that's a better diagram of what these are. So why are they important? Our team has thought passionately for 10 years now that these are as important as the DNA itself. But they've been very highly overlooked. The DNA has become very sexy in research. Billions and billions have been spent. And what our team predicted when we started has turned out to be true. The DNA is not getting there for early detection. Why? It's just too rare a marker. It doesn't matter how much superpower computers you put on it and sequencing and whiz-bang machines, if it's not there in the quantity to measure it, it's not going to work. And that's exactly the problem. They've just all, the big DNA companies that have 5, 10, 20 billion valuations all put together have worked out. So what is this? This is the nucleosome. It's 8 proteins and 9 proteins and a very wide range of epigenetic signalings on the tails, as well as combinations of all of these. There are hundreds and hundreds of markers on this, which are one of the very genesis of the cancer. Miswiring in these causes cells to continue to reproduce. So there's been billions and billions of spent on the DNA itself. We are the only company that has intellectual property. We've been around 9 or 10 years, so I'll cover that a little later as well, that has intellectual property on using these in circulation. So in the cell, you can diagnose things, but the problem is you've got to do biopsies and you have to know the cancer is there. So when the cells die, the body cuts up the chromatin in nucleosome-sized chunks to be reprocessed. And so cancer patients, which has been shown for about 15 years, have a lot more of these in the blood. Not only are there a lot more of them in the blood, but they have a lot of different signals on them which are different from ones in healthy nucleosomes. And why we can do it with just a drop of blood, not the 5, 10, 20 mils needed in the DNA companies, is the changes tend to be genome-wide, meaning millions of these are altered, not just a small number. So that means if you look at the DNA companies, they're looking for the needle in the haystack. We're looking for the haystack of nucleosomes. So we've got some very exciting technology and large trials underway, and I'll go through the potential markets. But basically, it's every adult person who can afford a blood test. And the cost of putting a test together, you're looking at a few dollars per assay. You put a few together, the cost of goods are below $10. And then you'd market it for $100, $150 in the US and about half that in Europe and about half that again in Asia. So very profitable, and we're not looking to charge very high prices. We want to make it very accessible worldwide, get something that really is ubiquitous. And the platform we're on is called the ELISA platform, which means it can be run in any lab on just about any piece of machinery. And there are dozens and dozens of different types of ways of running ELISA tests. Pretty much every test that you currently have, well, the very common ones, a majority of them are ELISA-based tests, which just means 2 antibodies. So again, the nucleosome. So this is, if you listen to a lot of people, they'll say it is a drop of blood. If you're looking for DNA, or if you're looking for circulating tumour cells, that is clearly just not the truth. If you are looking for something that's only a few hundred events in your entire system, you're not going to do it with a drop of blood. Just the physics of it all, it's not going to work out. You're going to get enough to measure. So to be a routine test, it has to be just a drop of blood, not a separate 5 or 10 mil draw. It has to go to one special bat cave in the middle of some planet, different place on the planet. It has to be run in every lab. Now, if it's not routine, you are not going to get compliance to 80 or 90% of people. If you give the people, this is kind of bad too for humankind's way of doing things, but if you give people a chance, a ton of people will duck out of it. If they have to do a special blood draw, if they have to go for a scan or a colonoscopy, all those things can be very effective, but huge numbers of people just don't do them. So we think we have the only way of getting there. Our intellectual property is very largely granted and extremely broad because the nucleosomes have been overlooked by every other group in the process of going through the DNA and other issues. So we have a huge growing patent family, a lot of which is granted, which in a lot of cases means we have all the markers on the nucleosomes for every living thing in every bodily fluid. It's massive and it's growing all the time. This is the very simplest plate format for ELISA. There's like 96 little test tubes. You don't need an entire test tube when you have 10 microliters, so it's shrunk down. And you run one little assay in each one. Extremely simple platform of 2 antibodies in these plates. That's how we can do it so cheaply. And we can do it so cheaply and quickly because the markers are so common in cancer patients for the reasons which I outlined. So given our test is incredibly broad in our platform in applications, it can be either a frontline test, which is the biggest market, which means everybody, asymptomatic if you don't have symptoms or other process. Usually by age, you tend to start 45, 50, 55. for a whole lot of reasons, health economics, which we won't go into, but it's usually not worth screening very young people. The biggest risk factor for every cancer, almost every cancer, is age, which we don't like to hear. We're all getting older, but the older you get, you're just much more likely to get cancers. And you can also use it in conjunction with other tests. You can use it as another diagnosis, and also for patient monitoring, all of which I think we can have some part in, because we have the intellectual property for all of this. This is no secret to everyone. Cancer is a massive problem, but it really does not have to be. If you have a cancer resected in stage 1 or stage 2 of something like colorectal, you have a 95% chance of living 5 or more years. If you're found late, which is all too many people are because they haven't got screened, your chance is about 5% of lasting 5 years. So that's not only terrible for yourself and your family, it's terrible for the health system, because they've got to spend a fortune keeping you alive, and chemotherapy and all those other things which happen. So, the best way to treat it is a blood test, and a truly routine blood test. Most prevalent cancers, most people know most of that. Of course, just what I said, the American Cancer Society saying screening is the best option. So just a quick one here. You're probably all, I guess even myself, I was not a scientist in this space. You have a general perception. You go to the doctor, you get a blood test for cancer. That's absolutely not the case. The only one currently accepted for screening in common use is the prostate. So the women out there, that doesn't include you. So basically women have no cancer, which is currently screened for by blood. There are some markers which are commonly used, but they're not very good and they're not approved for screening. Things like CEA, CA125. So the vast majority of other diseases are diagnosed through blood tests for the very obvious reasons, which if they can be a routine, easy to use one. Cancer tends to be diagnosed by x-rays, mammography, scanning, biopsies, colonoscopy. Almost all of those come down to the fundamental level of actually looking at the cancer through a scan or mammography, colonoscopy. Some of those can be accurate, but that involves complexity, it involves a lot of process and expense, and it means, again, people duck out of it. So they don't get screened. Some of them are very, very good. The colonoscopy is a fantastic test. If you're minded to do it, it's a great one to do. It's just really expensive. In the States, it's about $3,000 a person, and it's just it's not really efficient for screening, because about, again, 35–40% of people just refuse to do it, even when it's provided for them. So again, the only answer is a blood test. This is what I talked about before, participation. Again, just another way of saying who does the tests. Even in the most compliant societies on the planet, not to stereotype, but potatoes like Denmark, Holland, national database of everyone, they follow you up, free fecal testing, free colonoscopy, still they top out in the 60 to 70% range. Even in America, with the tens of billions spent on colon screening, 62–70% is where it tops out. So, the data from our tests, we had a platform which I showed you the lab, which we've been rejigging for the last 2.5–3 years to make sure it's robust, reproducible, and can be sent to any lab in the world. We had early data which showed between 80 and 90% area under the curve, which comes down to the accuracy. We've just finished the first few assays through the new process, which I'll get to. We've redone everything on the platform, from the antibodies to the control. We've changed from serum to plasma, and we've upgraded to magnetic beads for some of the platforms. The first assays through, we're getting extremely good accuracy, and we're in the process now of launching the other ones and running them through the trials. So we would expect in the colorectal space to be somewhere in the 80 to 85% accuracy range. Not as good as a colonoscopy. If you have a trained technician with a camera up inside you, they detect about 95% of them. But it's extremely good for a blood test. Order of magnitude, the PSA is not very accurate. It's in the region of 70%. So, and it's very commonly given because it's routine, easy to use. The doctor ticks a box and it goes away to the lab and comes back with information. If the PSA was $600 and 10 mils of blood, it just would never be used. So we're working, these are in colorectal and lung, but we've done other cancers as well. And as I said, we're running very large trials now in the thousands or tens of thousands worldwide in colorectal and lung, and we'll add others to it. Very heavily subsidized by the National Cancer Institute in the US. The US study, we're paying just over $2.5 million, just under $3 million, sorry. If we're paying 3,000 or 4,000 a patient, that would be $40 or $50 million. But again, everyone is really desperate for a blood test, so they've been extremely helpful, as has the Taiwanese government. We're working with some groups in China now, as well as several European trials. So very large numbers are needed to really show the efficacy. And that's all underway, and we'll have the first results starting next year through 21 on the big trials. and regulatory strategy. We intend to go through the FDA, the front door, do a big PMA study, do it right, do large trials, also large trials in Europe and Asia. Just because we have a very low cost test, we're getting heavily subsidised, we may as well do the large trials. You can often spend 2 or 3 years defending that it should work in China or Taiwan or in the United States. You may as well just do the trial when there's a small number of millions per trial. And we have, I think, well, I didn't mention at the start, but we have like $23, $24 million left. We're spending 3 or 4 a quarter. So we have more than enough money to get these through. As I said, just quickly, we're not veterinary specialists at all, so we're not interested in doing this ourselves. We found some very good vet school in the United States, Texas A&M, one of the best vet schools in the world. They live and breathe animal health. And it turns out, things which I found very surprising, but are actually true, there are many, many more dogs diagnosed with cancer every year than humans in the US. How can that be when there's less dogs? Pretty simple, actually, if you think about it. Dogs get cancer when they're 8 or 10 years old, not when they're 60 or 70 or 80. So it's a huge problem. Very hard to scan a dog or an animal because they don't stay still, so you've got to give them anesthetic, and the human proteins don't work. But nucleosomes, the epigenetic signals, are preserved through just about every living thing. Huge potential market. You charge about the same as the human market. We're just in Texas at the end of this month finalising the joint company with Texas A&M. And the other good thing in the dog market and the animal market is much smaller trials, more similar to pharma trials. 100, 200, 300 samples can get you to launch a product. So we're very happy with Texas A&M. They're absolutely fantastic in this space. And it's almost exactly the same conditions as in humans, so almost exactly the same assays themselves. It's really exciting and something which will become very serious through the end of this year and into next year, and potentially a multi-billion-dollar market by itself. And again, our IP covers every living thing. Just very quickly on this, it's a little more complicated. We've been labelling the antibodies, the samples in the plastic plates. We've also capturing the nucleosomes now and pulling them out, which means, if you remember from the slide, the very first slide, you get the nucleosome and the DNA. So we announced a few weeks ago, we can capture the nucleosomes and also target short nucleosomes, which have been shown to be much more common in cancer. So when I mentioned before, the DNA companies have real problems finding the needle in the haystack analogy. What this has the promise of doing is brushing away the haystack. If we can pull out the nucleosomes preferentially from the cancer, we're pulling out the DNA preferentially from the cancer. So that's in process, and we'll have a lot more data in the next coming months and quarters. And again, this really shows we are at the heart of epigenetics. The switching mechanisms on the nucleosomes are incredibly important in a range of things. If you think what's the difference between DNA and epigenetics, the genetics and epigenetics, think of it this way, a quick way to describe it. The DNA is like the hardware on a computer, the epigenetics is the software. And you're probably aware, where does the computer normally go wrong? It's in the software. And we absolutely dominate this field in the epigenetics using nucleosomes in circulation. And we've had a very strong belief over the last 10 years. We've been quietly in the background, swimming in our own lane, raising money, developing the platform. And it's been some ups and downs like any other company, but we're really confident now with our intellectual property, our team, and what we're doing is incredibly important. And the next few years should be really good for us. So I think the reason why the stock market's been roaring in the last few months, I think people are really realising the epigenetics is a big part of the story, and we're going to be a very big part of that story. So research kits, again, we just launched them with our partners in America and Active Motif. Not a huge revenue stream. It's not the multi-billion-dollar markets in humans and each cancer or dogs, but it's good to get researchers using it. It helps mankind. Also, if they discover something interesting, they've got to come back to us for a commercial license, if it's a company, which is a whole different discussion. I'll finish off with the most important thing. I think we've had 10 years as a company. It's a really long process developing a groundbreaking platform, and that's exactly what we've done. We have a team who've done an amazing amount of work. It's stuck together for the last 9 or 10 years to the situation we're in the hundreds of millions of dollars in value now. For example, our chief scientist worked for the World Health Organization and the NHS for 2 decades, developing ELISAs in different areas. Our chairman has been putting a ton of his own capital early on when we're developing from scratch, has been extremely helpful. We have a great team in Belgium. Again, that's where all of our research is done. We've gone for the lowest cost-based area, and it's been a great team. Belgian government's been very helpful, and it's been a great place to be. And a very strong scientific team, including Dr. Quay, who's running our Asian operations, and Nathan has just come on board for the vet side. He's a Texan. So just a quick summary again. Blood-based diagnostics, very, very breakthrough technology in nucleosomes. I think a time has come now. It's taken 9 years to become hopefully an overnight success as we really start to roll out our platform. We have the IP, we have the team, and absolutely epigenetics are incredibly important, and we have dominate the space because we've spent all this time developing what we have from scratch. That's Volition.
Thank you. Do you have any questions for Cameron, please? Again, if you do, raise your hand. Gentleman in the back here there, please, Chris.
Simple one. What's the false positive rate on the test?
It's so sensitivity and specificity. The AUC depends on where you draw the curve. So we say AUCs. The American market's much more determined to get every cancer, even if there's some false positives. Where in Europe, much more concerned with not having too many false positives. So the follow-up. So at AUC of 85 or 90, we normally would draw it somewhere in the middle. So about 85%, which is lower than something like a colonoscopy. Much better than a low-dose CT scan for lung cancer, and much better than the PSA, for example, which is in the region of 70, depending on how you do it. But the nucleosomes, which is such a simple platform, keeping it to $100 test in the US and less here, you are putting together 4 or 5 assays to keep that cost price down. But it'll be somewhere in the 80%, 85% range, we think, once we finish the trials. That's what we've been getting.
Does that mean, does that mean 1 in 5 get a false positive?
1 in 7. Yeah.
1 in 7. Okay.
Absolutely.
So just to kind of come on, if someone has a positive, you would expect them in the colonoscopy to go on to a further test?
Oh, yeah, sorry. So and depends on what the false positive is, but say in colon cancer, that means you go for colonoscopy. So every one of these is a triage, if you will, for the next level of tests. So in the US, they're not that concerned with false positives because everyone normally gets a colonoscopy anyway. European governments are much more concerned when you've got to pay for a colonoscopy that they're not paying for a ton of them. So the standard of care typically in Europe is FOBT or FIT, for example, in the colon space, the poop and scoops, if you will. They put the sensitivity, so specificity up to 90 or 92% and reduce the sensitivity, sometimes the FOBT down to 30 or 40%, just to get the highest specificity. But the Americans are the other way around. They prefer a higher cancer rate and more false positives. You never know when you're doing trials, but certainly there are hundreds and hundreds of markers on the nucleosome. We've only looked at a very small number, so we're doing a lot of discovery now. A lot of them, no one has any idea what they actually do and the purpose. So we would expect it's going to continue to get better and better. And with the trial process, once you've done it one time in a big PMA, you can do 510Ks. It's 500, not 10,000 in colorectal if you wanted to update it. So it's nowhere near as expensive to update the panels as the first one, because you use the first as a predicate to the others. So we'd strongly, you never know until you've done it, but there's every reason to very firmly believe that it would get better and better as time goes on. But anything in the 80%, 85, 90% range is an extremely viable product because the accuracy in that 30, 40, if targeting the non-compliant market first, which is in the US is 30, 40%, but if you're looking at parts of Asia, parts of Southern Europe, you're looking 80 or 90% of people don't get tested. It's horrible. So the only way of getting the people who aren't currently going now is through something where it's done with your normal blood work, like your cholesterol, all those things that happen. You'd have a new Q test, which is ours, based on all of that as well.
Okay, cheers.
And gentleman here, bring the microphone.
I just wanted to understand what the cost-benefit was of testing everybody with a blood test.
So yeah, absolutely right. So some cancers, it's worth it if there's a high prevalence. So colorectal, it's one of the more prevalent cancers, very treatable. The health economics have been proven with quite inaccurate tests like FOBT still to do it. You probably would not screen everyone for lung cancer unless it was very specific because it's a very deadly cancer, but much rarer than colorectal. So you would start with the high-risk groups. Smoking is obvious. In lung cancer, things like pancreatic, you'd start with people with adult-onset diabetes, which is still a very big market. But unless you were in the high 90s in the rarer cancers, for specificity, you would not screen everyone. But massive markets, you're looking at, even with the less common things like lung cancer and smokers, diabetes, you're looking at very large markets and the non-compliant markets for the ones you do. But there are only a small number of cancers you would screen everybody. because of the health economics in a socialised system.
Cameron, just a quick question. Can you compare your test to the GRAIL test, which is a blood diagnostic test for early diagnosis of cancer, a big trial, and just how their test compares to yours, and then also how the IP is separated? Thank you.
Absolutely. So GRAIL, Guardant, Freenome, there's about 4 or 5 groups who look for DNA, what we talked about earlier, which is actually looking for the genetic signal. They've actually all, Freenome broke ranks about a year ago to say that the DNA is not working in early stage cancers. So if you read all of those groups, and I'm not sure how their IP all separates, but they're all looking for DNA and it's not working. So they're going for methylation through sequencing, which is an epigenetic marker, but not nucleosomes. So what they were founded to do was the genetics, which has nothing to do with us. They're moving on to epigenetics through methylation and methylation alone at the moment. But we have every single structure on the nucleosome in circulation in every living thing, so they're not touching our IP. But from what we do for the nucleosomes and the epigenetic structures, it's 100% us. The only ones we don't have are the sequencing for things like methylation. But it's a much more, because it's such a rare marker, methylation and the DNA, they need 5, 10, 15 mils of blood. Guardant's 360 test is about $7,000, something like that, which, good on them, they made $100 million in revenue, very well-run company, great people, but that's not going to be a screening test in Europe or Asia or even probably the United States. And when you're looking for a very rare marker, you need 3-4 vials of blood, which means compliance goes through the floor, which means because there's such a rare marker, you have to have a lot of it. And it means you have to ship it to a bat cave and go through thousands of dollars of testing. It's not routine. So it's completely, I think both can thrive if they're more accurate but very expensive, and ours is a mass market test.
Cameron, thank you very much indeed.
Thank you.
Thank you. That's all we have time for from Cameron.